From the previous lesson, we learned that cells come in different sizes and shapes. In spite of their diversity, cells can be lumped into two broad types, the prokaryotes and the eukaryotes. Before we discuss cells in general, we will briefly describe a biological entity which has affected the whole world in ways we never imagined. Do you know what it is? It's the virus. We all know, based on our information on the coronavirus, viruses can reproduce and evolve. So are they considered living things too? Using the characteristics of living things we learned from our elementary science, let us see if viruses are alive. Living things must maintain homeostasis. Homeostasis is all about balance. Can something control its internal temperature or its internal contents? According to the cell theory, living things must be made of cells. Viruses are not made out of cells. A single virus particle is known as a virion and is made up of a set of genes bundled within a protective protein shell called a capsid. Certain virus strains will have an extra membrane, like a lipid bilayer, surrounding it called an envelope. Viruses do not have nuclei, organelles, or cytoplasm like cells do, and so they have no way to monitor or create change in their internal environment. This criterion asks whether an individual virion is capable of maintaining a steady state internal environment on its own. Though some have argued that the capsid and envelope help virions resist change in their environment, the general consensus is that viruses do not pass this first requirement for life. For this characteristic, the verdict is fail. Living things have different levels of organization. Smaller building blocks come together to make a larger product. Viruses certainly do this. They have genes made from nucleic acids and a capsid made of smaller subunits called capsomeres. In this case, the verdict is pass. Living things reproduce. Viruses definitely multiply. Viruses must use host cells to create more virions. Since viruses don't have organelles, nuclei, or even ribosomes, they don't have the tools they need to copy their genes, much less create whole new virions. Instead, viruses enter living cells and then hijack the host cellular equipment to copy viral genetic information, build new capsids, and assemble everything together. We use the term replicate instead of reproduce to indicate viruses need a host cell to multiply. And in this case, the verdict is maybe. Living things grow. They use energy and nutrients to become larger in size or more complex. Viruses manipulate host cells into building new viruses, which means each virion is created in its fully formed state and will neither increase in size nor in complexity throughout its existence. Viruses do not grow. Thus the verdict is fail. Living things use energy. Creating new virion units is a major undertaking, from building nucleic acids to putting capsids together that cost a lot of energy. However, all the energy that goes into this construction comes from whom? You guess it, the host. While viruses will definitely benefit from the use of energy, they are latching onto the host metabolism to get it. So the verdict here is maybe. Living things respond to stimuli. A response to a stimulus is defined by an almost immediate reaction to some change in the environment. While viruses don't change behaviors in response to touch or sound or light the way that humans, bacteria, or sea sponges might, there has not been enough research done to definitely say that viruses do not respond to anything. So the verdict is maybe or unknown. Living things adapt to their environment. Adaptation and evolution happen through unintentional changes, such as mutations, that are advantageous to an entire species. Viruses definitely adapt to their surroundings. HIV mutates quickly because it makes frequent mistakes while replicating its genome. Because the virus is constantly changing, it makes it very hard to design drugs and vaccines against it. One drug might prevent a large number of virions from replicating, but just a few will be unaffected. Those surviving virions will continue to infect more cells, making copies of the resistant strains. Thus, the verdict is pass. So let us summarize. Viruses are made up of smaller components but not made out of cells. Even they can't keep themselves in a stable state. They don't grow, and they can't make their own energy. They can definitely replicate, but can't reproduce and adapt to their environment. The majority opinion among biologists is that viruses are not alive and are not considered living things. Now let us go back in studying different types of cells. Let us first recall our taxonomy lessons from your elementary science. At first, there were thought to be only two domains, Eukarya and Prokarya, with five kingdoms, plant, animals, fungi, protists, and monera or bacteria. However, because of newly discovered species that can't be categorized in existing groups, we recognize today the three-domain and six-kingdom system. The three domains are Eukarya, which includes the kingdoms involving eukaryotic cells, Eubacteria, which includes the previously discovered members of the kingdom Monera, and the last domain, Archaea, which contains the Archaea bacteria or those that are tolerant of harsh conditions. Both Eubacteria and Archaea bacteria are considered as prokaryotes. The distinction between prokaryotic and eukaryotic cells represents the most fundamental division in the living world. Prokaryotes, more commonly known simply as bacteria, are a diverse group of primitive one-celled organisms. Prokaryotes are divided into two major groups, the Archaea and Eubacteria as mentioned earlier. Members of the Archaea bacteria are best known inhabitants of extremely inhospitable environments which include thermal hot springs, salty areas, and deep sea hydrothermal vents that can reach 110 degrees Celsius. The eubacteria, on the other hand, are widespread and live comfortably almost anywhere, in the air we breathe, in every surface, in nearly every drop of water, and even in our intestines. Some prokaryotes are our enemies, causing diseases such as tuberculosis, syphilis, and food poisoning. Other bacteria cause disease in plants, fungi, and other animals. Some prokaryotes are our allies, in decomposing remains, recycling nutrients, tidying up ecosystems, putting oxygen in ponds, and nitrates in soil. At first glance, the prokaryotes appear to be rather simple. Compared to eukaryotes, they have fewer internal parts and are generally smaller, averaging only about one-tenth the size of a typical eukaryotic cell. However, this is misleading. Now let us look closely at the structures of a typical bacterial cell. The first set of parts are those external to the cell wall. Glycocalyx is a highly charged layer of membrane-bound biological macromolecules attached to a cell membrane. This layer functions as a barrier between a cell and its surrounding. The glycocalyx exists in bacteria as either a capsule or a slime layer. The difference between a capsule and a slime layer is that in a capsule, polysaccharides are firmly attached to the cell wall, while in a slime layer, the glycoproteins are loosely attached to the cell wall. Flagella are filamentous protein structures commonly found in bacteria. They are typically used for motility or to propel a cell through liquids such as in bacteria and sperm cells. There are three main components to the bacterial flagellum. The filament, a long thin appendage that extends from the cell surface. The hook, this is a curved coupler that attaches the filament to the flagellar motor. And the basal body which provides the rotation. Fimbriae enable the bacteria to bind or adhere to specific receptor structures and thereby to colonize specific surfaces. Pili are used for motility. Fimbriae are bristle-like short fibers that occur on the surface of bacteria. Pili are long hair-like tubular microfibers-like structures present on the surface of bacteria. The next is cell wall. The cell wall is responsible for the shape of the cell, and it protects the membrane and the cell interior from adverse changes of the outside environment. It prevents bacterial cells from rupturing when the water pressure inside the cell is greater than the outside of the cell. It also serves as a point of anchorage for the flagella. The major component of the bacterial cell wall is peptidoglycan. Peptidoglycan is a polymer consisting of sugars and amino acids. Gram-positive bacteria have a thick peptidoglycan layer and no outer lipid membrane, while gram-negative bacteria have a thin peptidoglycan layer and have an outer lipid membrane. Because of this, gram-negative bacteria are more resistant to antibodies and antibiotics than gram-positive bacteria because they have a largely impermeable cell wall. The bacteria responsible for multi-resistant Staphylococcus aureus or MRSA and acne are examples of gram-positive bacteria, while those responsible for Lyme disease and pneumonia are examples of gram-negative bacteria. The next set of parts are those internal to the cell wall. Homeostasis is essential to the survival of a cell. One of the structures that is primarily responsible for homeostasis is the plasma membrane. The plasma membrane is a thin, flexible boundary between a cell and its environment that allows nutrients into the cell and allows waste and other products to leave the cell. All prokaryotic cells and eukaryotic cells have a plasma membrane to separate them from the watery environments in which they exist. A key property of the plasma membrane is its selective permeability, by which a membrane allows some substances to pass through while keeping others out. Control of how, when, and how much of these substances enter and leave a cell relies on the structure of the plasma membrane. The plasma membrane is composed of a phospholipid bilayer, in which two layers of phospholipids are arranged tail to tail. Moving with and among the phospholipids in the plasma membrane are proteins, cholesterol, and carbohydrates. Proteins at the inner surface are called integral proteins, When found on the outer surface of the plasma membrane, they are called peripheral protein. Other substances in the membrane, such as glycoproteins and glycolipids, stick out from the plasma membrane. Plasma membrane will be discussed more in detail in Module 5. Cytoplasm refers to the material or protoplasm within a living cell, excluding the nucleus. It is the semi-fluid substance of a cell that is external to the nuclear membrane and internal to the cellular membrane, sometimes described as the non-nuclear content of protoplasm. The liquid portion of the cytoplasm is called the cytosol. The nucleoid, which means nucleus-like, is an irregularly shaped region within the cell of a prokaryote that contains all or most of the genetic material. A bacteria contains two forms of DNA. The chromosomal DNA is a single long continuous thread of double-stranded DNA. The other form, plasmids, are circular double-stranded DNA molecules that usually carry genes for antibiotic resistance or enzyme synthesis. In contrast to the nucleus of a eukaryotic cell, a nucleoid is not surrounded by a nuclear membrane. Ribosomes are tiny structures responsible for protein synthesis. All prokaryotic and eukaryotic cells have ribosomes but differ in their subunits. Lastly, inclusions serve as reserve deposits that contain excess nutrients. Then what makes prokaryotes appear simple? Prokaryotic cells have virtually no membrane-bound organelles. They have no nucleus, which is the large central structure found in eukaryotic cells. Their genetic material is within the cell's internal fluid in a region known as nucleoid. Numerous ribosomes necessary for protein synthesis reside within their cytosol, the cell's fluid compartment.